Hello everyone. My name is Snehlata and I'm a PhD student and a PMRF fellow in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today we will continue with the next lecture of the chapter Biosynthesis in Higher Plants. So in this lecture we will start from where we left in the last lecture. So what we are studying in the last lecture was about an experiment which told that about one experiment that was performed by T.W. Eagleman. And what he told, he described that what wavelength of light is important for photosynthesis. And we saw this thing in the last lecture. So what we saw, that he used a green algae named Cladophora as his model organism on which he did the experiment. And he used a prism. This is a prism that split the white light. So this is the light. So he used the prism to split the light into the rays of different wavelengths. And what he saw, he observed that he kept a cladophora in a solution in which there were other aerobic bacteria present. So the the red color here is representing the aerobic bacteria. And what he observed that after some time, when this light of different wavelengths, when they fall onto the different region of this cladophora, he found that there was accumulation of the aerobic bacteria in certain regions. And what would those certain regions? These certain regions are represented by these green dots here. And these are those regions in the cladophora where the light of a wavelength corresponding to the blue ray and red ray falls. So from this, he concluded that the blue and red wavelength of light is important for photosynthesis. And the reason why? Because we know that during the photosynthesis, oxygen is formed as byproduct. So if there is increase in the amount of photosynthesis, there will be increase in the concentration of the oxygen that will be formed, right? And this increase in the concentration of oxygen will lead to the increased accumulation of aerobic bacteria. Why? Because the aerobic bacteria are the one that requires oxygen for their survival, right? So because of this reason, we can see that there was accumulation of bacteria at different region of the cladophora, and that region corresponds to the blue rays light and red rays light. And hence this blue and red wavelength light is important for photosynthesis. Apart from this, he also described about one concept in the photosynthesis, and that is known as action spectra. So what is action spectra? So action spectra basically tells that wavelength, it tells about wavelength at which maximum photosynthesis takes place. Okay. So it tells about the wavelength at which maximum photosynthesis takes place. Okay. So these two experiments are connected, right? So basically the wavelength at which maximum photosynthesis will happen is the wavelength at which maximum oxygen production will take place through photosynthesis, right? So here we draw this spectra between two axes. The one axis was oxygen concentration and the second axis was wavelength. And this, what he observed, so this wavelength will be from violet to red. So suppose this is VI, B, G, Y, O, R, right? So these are the wavelength of different light. So when he draw the spectra, this action spectra, he observed that the maximum oxygen production was like this, or the oxygen concentration curve was like this. So the oxygen concentration was higher at blue wavelength light and at red wavelength light. So this is the action spectra, and this action spectra is of what? So this is the action spectra of the chlorophyll A. So this is the action spectra of chlorophyll A. Okay. So chlorophyll A is a pigment about which we'll study in upcoming slides. But for now, so because if you remember in the last lecture, we saw that there are various components that are required for the photosynthesis, among which chlorophyll was one of the component, right? So this is a chlorophyll that actually absorbs this light, or the pigments that are present in the plant leaves. These pigments actually absorb the light and leads to the photosynthesis. So if you talk about the action spectra, so the action spectra will be of the pigments. And here the action spectra that I have drawn is of the chlorophyll A. Okay. Okay. So this was one experiment that proved that what wavelength of light is important for photosynthesis. Now, there are certain other experiments, among which the first experiment is the that determines the source of oxygen during photosynthesis. So now we know that the oxygen comes from water, but back in 1800 or 1900, we are not knowing this thing. So at that time, the various experiments were performed to determine that oxygen comes from water molecule. So we know why this confusion was there, because if you remember the reaction of photosynthesis, it looks like this. It looks like this, right? Now, the question is, since the carbon dioxide also have oxygen and the water molecule also have oxygen, so this oxygen, from where it is coming from? So whether it is coming from this water molecule or this is coming from carbon dioxide. So how to prove this? Because both of these molecules are oxygen molecules, right? So for this, a number of experiments were performed. So among which the first experiment was performed by a scientist named C.V. Van Neel. Okay. So C.V. Van Neel, he is a microbiologist. So C.V. Van Neel is a microbiologist, which means he studied about the microbes, right? So what he did, he used a model organism named, he used purple sulfur bacteria. He used purple sulfur bacteria. Okay. He used purple sulfur bacteria. What is purple sulfur bacteria? It is a photosynthetic bacteria. Okay. So instead of using plant, he used a bacteria because he's a microbiologist. He was a microbiologist, so he used to work on microbes such as bacteria. Okay. So he used a purple sulfur bacteria, and this bacteria is a photosynthetic bacteria. So it means it does photosynthesis, right? So, but this photosynthetic bacteria, in this photosynthetic bacteria, the photosynthesis reaction that takes place looks like this. So it looks like this. Okay. It looks like this. So what is the difference that we are observing here? The difference that we are observing here is instead of H2O, instead of this H2O, this bacteria use H2S. Okay. This bacteria use H2S. And hence the byproduct that was formed is along with the glucose, the byproduct that is formed here is what? Sulfur. The byproduct form is sulfur. So from this, it was proven, because in this case, when instead of H2O, H2S was used, it was found that the byproduct is sulfur. So from here, it was concluded, or it proves that the oxygen that is forming as a byproduct during photosynthesis, it is coming from the water molecule instead of carbon dioxide, right? Because if it would not have been coming from the water molecule, in this case also, in this purple sulfur bacteria also, oxygen would have been formed as a byproduct, right? So this experiment proved what? It proved that oxygen comes from H2O or water, not carbon dioxide. But in this case, also the photosynthesis is happening, right? But this photosynthesis is called, the photosynthesis that is done by purple sulfur bacteria is called as anoxygenic photosynthesis. Anoxygenic photosynthesis. Okay. Why? Because in this case, no oxygen is being formed. No oxygen formation is taking place. Right. So from this experiment, C.V. Van he proved that the oxygen is coming from water molecule, not from the carbon dioxide molecule. Okay, so this is the one experiment. Now, the second experiment was performed by another scientist named Robin and Hill. So Robin and Hill, because this was proven in the previous experiment was proven in the bacteria, right? So the another experiment was performed by Robin and Hill, and they worked on angiosperms. So what are angiosperms? Angiosperms are higher plants, right? So they are higher plants. So these scientists worked on angiosperms, and they used a plant named Stellaria media. Stellaria media. Okay. And I will not go into the detail of the experiment, but from this experiment, they proved that the oxygen comes from water molecule. Comes from water molecule. So the important point here is that they used a plant named Stellaria media, and these scientists worked on the angiosperm. So these are the scientists that proved that in the case of plant, also the oxygen comes from water molecules. Then came the third group of scientists named Rubin and Kamin. So what does Rubin and Kamin did? So they worked on chlorella. Okay. They worked on chlorella. So all of these scientists, they use different model organisms. Okay. Someone worked on bacteria, purple sulfur bacteria, then angiosperm, then chlorella. Okay. And what is chlorella? Chlorella is unicellular algae. Unicellular algae. Okay. So Rubin and Kamin, they worked on chlorella, which is a unicellular algae. And what technique did they use? So the technique that they use to determine that the oxygen comes from water, not from the carbon dioxide, is called as radioactive labeling. So they use a technique known as radioactive labeling. So what is radioactive labeling? So you must have heard about the isotopes, right? The isotopes. So these isotopes, they have different molecular weight, right? They have same atomic number, but different molecular weight. So same atomic number. Isotopes are what? The elements having same atomic number, but different molecular weight. Different molecular weight, right? For example, in the case of carbon, if you say carbon, so carbon we have naturally occurring carbon is carbon 12, but we also have carbon 13, right? And carbon 14. So this all have this atomic number of 6, but the molecular weight is different. So this, because it is 14, this will be heavier one, right? This will be heavier. So the Rubin and Kamin used this technique of radioactive labeling. And what they did with this radioactive labeling, they labeled oxygen of water with O18 isotope. With O18 isotope. So like carbon, oxygen also has different isotope. Naturally, it is present in the form of oxygen. So naturally, oxygen is present as O16, right? But it has other isotopes as well, that is O18. Okay, so of course it has if it has higher molecular weight, it will be heavier. Okay, so these scientists, they labeled the oxygen of water molecule with O18 and with O18, and the carbon dioxide, they do not label with anything. Okay, so the oxygen of carbon dioxide was not labeled with anything. So the carbon dioxide will have O16 carbon. Okay, now what they did when they performed the experiment, or when they performed this, kept this chlorella. So this is a solution having chlorella. Okay, so it has chlorella in here. Apart from this, they supplied it with the normal CO2. They supplied with the normal CO2 and radio-labeled water molecule. Radio-labeled water molecule. And then when they did the photosynthesis, so in the presence of light, photosynthesis will happen, right? Then they analyzed the oxygen. They analyzed oxygen formed. So if the photosynthesis will happen, the oxygen molecule will form, right? So they analyzed the oxygen form, and what they found, they observed the oxygen that was formed was O18. So oxygen formed contained O18 isotope. Okay. So from this, what they found? So what they did was they radio labeled the oxygen of the water molecule while the carbon dioxide was as it is. So it was as it occurs naturally. Okay. So the carbon dioxide was natural while the water or oxygen was radio labeled. Then they did the photosynthesis and analyzed the oxygen that is formed during that photosynthesis process. And what they observed that the oxygen that was formed, it contained O18 isotopes. And from this, it was proved that oxygen comes from water. Because if it would have come from carbon dioxide, the oxygen that is formed here will have normal isotope, right? It will have O16, but this was not the case. Okay. So from this, it was proven that oxygen comes from water rather than carbon dioxide. Okay. So with this, we have completed all the early experiments that were performed to prove that what are the factors or the components that are required for the photosynthesis process. Now let's start with the actual photosynthesis process, or what are the different things that happen during the photosynthesis. So first of all, let's talk about the site of photosynthesis. So what is the site of photosynthesis? So if you start, one second. So the site of photosynthesis, if you start, the photosynthesis happens in plants, right? It happens in plants. But in plants, where does it happen? In plants, it happens in leaves, right? Now, in leaves, what do we have in leaves? In leaves, we have cells. So what kind of cells does perform photosynthesis? So in leaves, the mesophyll cells. The cells that perform photosynthesis are mesophyll cells. Okay. Now, inside the mesophyll cells, because we have studied in the cell chapter that cells consist of different organelles. So where does the photosynthesis take place in the cell? So in the cell, the photosynthesis takes place in the chloroplast. So what is chloroplast? Chloroplast is a organelle. So this is what? This is a organelle. Now, inside the chloroplast, where does the photosynthesis take place? So because a chloroplast is also double membrane structure, right? We'll see the structure after this. So in the chloroplast, where does this photosynthesis takes place? So it takes place in the place called stroma. Takes place in place called stroma and thylakoids. Stroma and thylakoids. Okay. So this is the site of, one second. Okay, sorry. Okay, so this thing I have to write here, I'll quickly write. So the site of photosynthesis will be, first it will take place in plant, then from plant it will have in the leaves, then in the leaves where in the mesophyll cells, it will be in the mesophyll cells, then in the mesophyll cells there is a special organelle known as chloroplast, and in the chloroplast it takes place in stroma and thylakoids. Stroma and thylakoids. Okay, just a second. Okay, so this is the site of the photosynthesis. So if someone asks about the organelle of the photosynthesis, the organelle will be chloroplast. And if someone talks about the cell, so cell is mesophyll cells. Okay. Now let's look at the structure of the chloroplast. So in the chloroplast, what all things we have? We have the chloroplast is double membrane structure. So it has one outer membrane and one inner membrane. So it is double membrane structure. It is double membrane structure. Okay. Now, apart from this, these chloroplasts, they have a stack-like structure, among which a single unit of the stack is known as thylakoid. So this is known as thylakoid. And the stack of this thylakoid, this entire structure, the stack of this thylakoid, this is known as what? This is known as grana. This is known as grana. So grana is what? Grana is stack of thylakoids. Grana is stack of thylakoids. Okay. Then these thylakoids are connected by another membranous structure, and this structure is known as stromal lamellae. This structure is known as stromal lamellae. Okay. Apart from this is the matrix. This is a matrix, or you can say stroma for the matrix, or it is also known as stroma. It has matrix of stroma. Okay, so this is the structure of chloroplast. Now, this chloroplast, if you see, it has two types of structure. The one is a matrix, right? So the one is the matrix. The chloroplast, if you look at the structure, it can be clearly differentiated or distinguished in two types of two structures. The first one is the stroma, and the second is the membranous structure. Second is the membranous structure, because we can see that there are a large number of membranes present in the chloroplast structure, right? So what things do we have in stroma and in the membranous structure? So in the stroma, we have enzymes. We have different enzymes that we'll see in upcoming slides. Then in stroma, a reaction of a photosynthesis known as dark reaction. So it is a type of reaction in the photosynthesis that takes place. So the dark reaction takes place in the stroma. Apart from this, the stroma has DNA, because in the cell chapter, we have studied that chloroplast and mitochondria are the two organelles that consist of their own DNA, along with the nuclear DNA. In a cell, chloroplast and mitochondria consist of DNA. So this stroma or the matrix have DNA and ribosomes, because they also perform their own protein synthesis. So the stroma consists of enzymes. It is a site of dark reaction, and it has DNA and ribosomes. Now, what do we have in the membranous system? So first of all, the membranous system, what does it consist of? So if you look at it, we have thylakoid here. You can see we have thylakoid. Thylakoid has its membrane, and inside it has this lumen, right? So the membranous structure consists of thylakoidal membrane. It consists of thylakoid membrane plus it has stromal lamellae. This stromal lamellae is also membrane, right? So the membranous structure consists of thylakoid membrane and stromal lamellae. Okay. And this is the site for the membranous structure is a site for light reaction. It is a site for light reaction, and it contains pigments. Membranous system contain pigments such as chlorophyll, Okay. So the chloroplast, it is divided into two different compartments. You can see the first one is the stroma, that is the matrix, and the second is the membranous structure that consists of the membranes. And in membranes, it is mainly consists of thylakoid membrane and the stromal lamellae. And this is the site of light reaction. Now, one thing that we can notice here is that the entire photosynthesis is not taking place at one site. So in the chloroplast itself, there is a division of labors, right? Because the light reaction is taking place in the membranous structure, while the dark reaction of the photosynthesis, it is taking place in the stroma. So there is the division of labor in the chloroplast. So we can write in chloroplast, there is division of labor. So what do I mean by division of labor? By this, I mean that it has different sites for light and dark reaction. It has different sites for light and dark reactions. Okay. So this is about the chloroplast. Okay. Now, what are the reactions? I was talking about the light and dark reactions. So basically, what is light and dark reaction? So what are the reaction that happens in the case of photosynthesis? So the reaction that happens in the photosynthesis are of two types. Okay. The reactions in the photosynthesis consist of two types. So there are two types of reactions. Two types of reactions that occur in photosynthesis. And what are these two types of reaction? The first type is the light reaction. The first is light reaction. And the second is dark reaction. Okay. Now, what is the difference between the light and dark reaction? Now, let's see. The first difference is the location. So, as we saw, the light reaction, it takes place in the thylakoid. It takes place in the thylakoid and stromal lamellae. It takes place in the thylakoid and stromal lamellae, while the dark reaction, it takes place in the stroma or the matrix. Stroma or matrix. So this is the location of the light and dark reaction. Now, why the light reaction is named so? Meaning why it is called as light reaction? Because it is directly dependent on the light. So light reaction is directly dependent on light. Okay, so it directly depends on the sunlight. While the dark reaction, it is not directly dependent on the light, but it depends on the product of the light reaction that is formed. Okay, so the dark reaction, one important point, dark reaction does not mean that it takes place during the night time. It can take place anytime, but it depends on the product of the light reaction to happen. Okay, so it does not, we can write that it does not depend on the light directly. Does not depend, the dark reaction does not depend on the light directly, but require products of light reaction. Require products of light reaction. Okay, it requires products of light reaction, and hence it is known as dark reaction. But dark reaction does not mean that it happens during night time. So it does not mean that it happens during night. Okay, it simply means that it does not directly depend on the light, but it requires the product of light reaction. Okay, now what is formed during the light reaction? What are the products that are formed during the light reaction? So during light reaction, ATP and NADPH are formed. ATP and NADPH are formed. Okay, and these ATP and NADPH, these are known as assimilatory power. These are known as assimilatory powers. Okay, now what is formed during the dark reaction? So during the dark reaction, glucose is formed. Glucose is formed, but we know that this glucose is not stored in its original form, but it's get converted into starch. So glucose is formed, which gets converted into starch during the dark reaction. Now, during the light reaction, what things are important? So in the light reaction, we need, or the plant need, the pigments. So it requires, light reaction require pigments or light harvesting complex. So we'll see in the upcoming slide what is light harvesting complex. But this light harvesting complex is required during the light reaction. Now, what is required during the dark reaction? So in the dark reaction, it is an enzymatic reaction. So it requires enzymes, right? It is an enzymatic reaction. And what enzymes that is required? It requires rubisco. It requires rubisco. Okay. So rubisco is an enzyme that catalyzes the formation of glucose, and this glucose formation takes place in the dark reaction, right? So this is the difference between the light reaction and dark reaction. Okay. Now, let's see the position of chloroplast. So position of chloroplast, the position of chloroplast in the that is present in the mesophyll cells, it changes. Okay. So the position of chloroplast, it changes based on light intensity. Based on light intensity. Okay. So there can be two situations, like in the morning or evening, we have a bright light, but its intensity is not very high. So what happens? Like during morning and evening, we have moderate light intensity, right? We have moderate light intensity. Now, during this time, if you observe a leaf and if you look at its chloroplast, what kind of arrangement will we see? So during morning and evening, suppose this is a mesophyll cell. So during this time, your chloroplast, it will be located at the periphery. It will be located something like, it will be aligned something like this. So it will be at the periphery. Now, the reason why it is located at the periphery in the morning and the evening, because these chloroplasts are the one that are absorbing the light, right? And during the morning and evening, we have moderate light intensity. So the chloroplasts will be aligned in such a way that they can absorb maximum amount of light, right? So during this time, the arrangement of chloroplast is what? Peripheral, or it is located at the periphery, right? And why? So it is to absorb maximum amount of light. Maximum amount of light. Okay. Now there's second situation. So this is the first situation. Now there's second situation. You can say during afternoon. So what happens during afternoon? The light intensity is very high. So here the position is based on the light intensity. I'm not talking that it is during the morning or evening, but it is based on the light intensity. Even if the afternoon, if the light intensity will not be, if there's no intense light intensity, the arrangement of the chloroplast will be like this only. But in most of the cases, what happens? In most of the cases, during afternoon, the light intensity is very high. Light intensity is very high. So what happened during this case? So during this case, your chloroplast arrangement will look something like this. So the chloroplast will be arranged like this. So they will come to the center. Okay. So when the light intensity is very high, the chloroplast will arrange in the center. Arrange in the center of the cell. Center of the cell. And why is it so? Because when the light intensity is very high, there will be high chances of photooxidation. Now, what is photooxidation? Because of very high intensity, there will be the oxidative radicals formation takes place that can damage the chloroplast. Okay. So to prevent this arrangement is to prevent photooxidation. To prevent photooxidation, or in simple term, you can say to prevent damage of chloroplast. To prevent damage of chloroplast. So this is the position of the chloroplast based on the light intensity. When it's moderate, they will be on the periphery to ensure that they absorb the maximum amount of light. But if they are when the light intensity is very high, they will be low, assemble in the center so that it can prevent the photooxidation or damage of the chloroplast. Okay. Now, yeah, so yes. Okay. Now we'll talk about the pigment. But before that, we should see one question. So suppose if someone asks you that if the sunlight or the light source goes off, then what will be the first reaction to get to stop? So if the question is if sunlight or you can say light source or light source goes off, what will be the what will be the first reaction to stop? First reaction to stop. What will be the first reaction to stop? Will it be light reaction or dark reaction? So in this case, what will be the answer? The answer will be light reaction. Light reaction, because the dark reaction is dependent on the product of the light reaction, right? So the ATP and NADPS that is formed, it can get consumed anytime by the dark reaction. So if the sunlight goes off, the light reaction will immediately stop. So this light reaction will immediately stop. But the dark reaction, it will goes on until this product that is formed by the light reaction, that is ATP and NADPH, gets consumed. Okay. Okay. Now let's talk about the pigments. So we saw that these pigments are the part of the are important for the light reaction. But what actually the pigments are? So by pigments, till now we have studied that it means color. So they contain certain colors. But from where this color comes from? So if we have to define the pigments, we can define it as a chemical compound. Pigments are the chemical compounds. These are the chemical compounds that can excite its electron. That can excite its electron at a specific wavelength of light. At a specific wavelength of light. At a specific wavelength of light. Okay. So what is pigment? So suppose this is a pigment. This is a pigment. So if it receives a light ray, its electron will get excited and it will move to the excited state, right? So it will have an excited electron. So electron gets excited, and then from its excited state, it will come back to the ground state. So this electron, after some time, it will slowly come back to the ground state. But when it starts coming to the ground state, it will start releasing energy. Okay. So this electron release energy, and this energy can be in the form of light or heat. It can be in the form of light or heat. Okay. So, and the light that it will release will have higher wavelength. The light that it will release will have higher wavelength. Okay. So this is what a pigment is. And this light, because of different wavelength, it will give color. And hence we say that the pigments are colored compounds. Okay. But these are the compounds that have the ability to get excite its electron upon receiving a light of a specific wavelength. And after excitation, when it comes to the ground state, it releases light of a higher wavelength, and this gives color. This gives different color. Okay. Okay. Now the question is how these pigments work. So in the case of photosynthesis, if we talk that the pigment that are present in the photosynthesis, so how do they work? So this pigment, the electrons in the pigments after excitation, what they do? So electrons in pigments after excitation, what they do? They can either transfer their energy to the nearby other pigments. Okay. So they can transfer energy to the other nearby pigments. Other nearby pigments. Okay. So suppose this is pigment 1, this is pigment 2, this is pigment 3, and this pigment 1 is receiving a light. So its electron will get excited. Now it will transfer its energy to the nearby second pigment, and then it will transfer its energy to the third pigment. Okay. So this electrons in the pigments after excitation, it can transfer energy to the other nearby pigments. Okay. But the pigments to which it will transfer the energy will always be of higher wavelengths. So these pigments will always be of higher wavelength. Okay, this will always be of higher wavelength. Now, what is the second thing that this pigment can do? So this pigment, after its electron excitation, can help in the enzyme catalysis. Okay, so it will help to perform a chemical reaction. To perform, you can say it helps in performing a reaction with the help of the enzymes that are attached to this pigment. With the help of the enzymes attached to this pigment. Okay, so the pigments that are the photosynthetic pigments or the pigments that are involved in photosynthesis can have two functions when they receive a light. The first is they transfer the energy to the other nearby pigments, and those pigments should always be of higher wavelength. And secondly, this pigments also have certain enzymes attached to it. So after excitation, they can also help to perform an enzymatic reaction with the help of those enzymes that are attached to this pigments. Okay. Now, what are the types of pigments that are present in the photosynthesis? So there are basically two types of pigments. The first one is the lipid soluble pigment. So the first type of pigment in photosynthesis is lipid soluble pigment. And among this, it is of 5 types. So the first one is chlorophyll A. The first type of the pigment is chlorophyll A, which is also represented as CHLA. Okay. Now, this pigment is present in almost all the plants. So present in all plants. Okay. So these pigments are present in all plants, but apart from plants, the pigments are also present in certain bacteria. Okay. In bacteria like blue-green algae. So what is blue-green algae? It is cyanobacteria. Blue-green algae is also known as cyanobacteria. And these are what? These are photosynthetic bacteria. And these bacteria also consist of chlorophyll A. Okay. So apart from the plants, it is present in all the plants. But apart from plant, it is also present in bacteria like blue-green algae or cyanobacteria. Okay. Then the second type of lipid soluble pigment that is involved in photosynthesis is chlorophyll B. The second type is chlorophyll B, and it is represented by CHLB. Now, where is CHLB present? So it is present in higher plants. Okay, chlorophyll B is present in higher plants and in certain lower plants such as Euglena and green algae. Euglena and green algae. Okay. So basically, this Euglena and green algae, although these are lower plants, but they have higher plant-like characters. So they have characters like higher plant. So this Euglena and green algae have characters like higher plants. Okay. So there's chlorophyll A, chlorophyll B. Now, third is chlorophyll C. Third pigment is chlorophyll C. Now, chlorophyll C is represented by CHLC, and these are present in diatoms, dinoflagellates. So present in diatoms and dinoflagellates. Okay, diatom and dinoflagellates. Okay, then fourth is chlorophyll D. Chlorophyll D, represented by CHLD, and these are present in red algae. So we know we have green algae, brown algae, red algae. So the chlorophyll D, these are present in red algae. Okay. Now the next one is carotenoids. The next lipid soluble pigment is carotenoids. So carotenoids, these are also called as accessory pigments. These are called as accessory pigments, means they help the other pigments. Okay. So these accessory pigments or carotenoids are of two types. Okay. So these are of two types. The first one is known as xanthophylls. The first one is known as xanthophylls, and the second one is known as carotene. Okay. So the carotenoids are known as accessory pigments, and these are of two types, xanthophylls and carotene. So in the lipid soluble pigments, we have five types: chlorophyll A, B, C, D, and carotenoids. Apart from this, we have water soluble pigment, or we can also call them as lipid insoluble pigment. Okay. So these are either, or we can say, or water soluble or lipid insoluble. In this, we have only one type that we have to study, that is phycobilins. So the water soluble pigment present in photosynthesis is phycobilin, and these phycobilins are of two types. The first one is phycoerythrin. The first one is phycoerythrin, and the second one is phycocyanin. Phycocyanin. So what is phycoerythrin? So this is red in color, and the phycocyanin, it is blue. It gives blue color. So by the color here, what I mean is, like I said, that the pigment, these pigments, what they do, they, when they absorb light, their electron get excited, and when these excited electron come back to the ground state, they release certain energy and a light, and this light is of higher wavelength. So this wavelength of light correspond to certain colors, right? So if I'm seeing that here the phycoerythrin is of red color, it means that upon excitation, when the electron of phycoerythrin come back to the ground state, it releases a light that corresponds to the red wavelength light. Okay. So by this way, what I mean that these pigments are different colors. Okay. Now, among these, okay, so these are like we have studied about the six pigments, but out of this, in the case of higher plants, there are only three pigments that are present. So what are these three pigments? The first one is the chlorophyll A. So in higher plants, chlorophyll A is present, chlorophyll B is present, and carotenoids is present, and carotenoids. So these are the three pigments that are present in the higher plants. So we will study about these pigments in detail. Okay, so the first type of pigment is chlorophyll A. So what is chlorophyll A? The chlorophyll A, it is a primary pigment. It is the primary pigment. It is present in the higher plant. Okay, and it is the most abundant pigment. It is the most abundant pigment. And what is the formula of chlorophyll A? So the chemical formula of chlorophyll A is C55H72O5 N4 Mg. Okay. So how does structure looks like? So the structure of chlorophyll, it consists of two parts. The first part is known as porphyrin head. So it has two parts. The first part is porphyrin head. Now, what does porphyrin head consist of? It consists of a central non-ionic Mg, magnesium element. So it has magnesium in the center, and then it has four porphyrin rings. Okay, so it has 4 porphyrin rings. So this is 1, this is 2, this is 3, this is 4. So these all 4 rings are connected to each other and they have nitrogen group. Sorry, nitrogen atom in them. So this has one ring, the second ring, third ring, and fourth ring. So these are connected to each other. And because of this, so these are connected to each other like this. And because of this, because it has this pyrrole ring, it is also known as tetrapyrrole. It is also known as tetrapyrrole. Okay. So in the porphyrin head, we have a magnesium. And this magnesium is what? It is non-ionic. So it will not be in the form of Mg2 plus, but it is in the form of Mg. So we have a magnesium in the center, and then this magnesium is surrounded by 4 pyrrole ring. And because of this reason, it is known as tetrapyrrole. Okay. And this together constitutes the porphyrin head. Now, if you look at the numbering will of the this pyrrole ring starts from here 1, 2, 3, 4. Okay. Now, from the fourth pyrrole ring, we'll have a long hydrocarbon chain. So it has a long hydrocarbon chain. So the fourth ring, from the fourth pyrrole ring comes a long hydrocarbon chain, and this chain is known as phytol tail. It constitutes phytol tail. So if you look at the chlorophyll structure, we have two regions, a porphyrin head and a phytol tail. The porphyrin head consists of a magnesium along with the tetrapyrrole ring, while the tail region consists of a long hydrocarbon chain that is called as phytol tail. Okay. Okay. So, and what color does this chlorophyll A gives? So it gives blue-green color. So chlorophyll A gives blue-green color. It gives blue-green color. Okay. Now this is chlorophyll A, which is the most abundant. The second type of chlorophyll that is present in the plant is known as chlorophyll B. So the chlorophyll B, it is green yellow in color. So this chlorophyll A is blue green color. Chlorophyll B is green yellow in color. Okay, and its formula is slightly different from chlorophyll A, that is C55H70 O6 and 4 Mg. So from the structure we can see we have 4 nitrogen, hence it has N4, and we have 1 magnesium, that's why it has Mg. Apart from this C55H70O6, it comes from the hydrocarbon chain as well as the tetrapyrrole ring. Okay. Now, what is the difference between chlorophyll A and chlorophyll B? So just by looking at the formula, we can see that in chlorophyll A, we have 5 oxygen, right? While in chlorophyll B, we have 6 oxygen. Now, the question is, what group actually constitute this difference? So, if you look at the difference between chlorophyll A and chlorophyll B, difference between CHLA and CHLB, what difference can we see? So we can see that in chlorophyll A, in the case of chlorophyll A, at the second pyrrole ring. So this is your first pyrrole ring. This is your second pyrrole ring. So at the second pyrrole ring of the chlorophyll A, we have CH3 group. So at second pyrrole group or pyrrole ring, at second pyrrole ring, we have CH3 group or methyl group. Okay. While in the case of chlorophyll B, at second pyrrole ring, we have CHO group. So now we can clearly see from where this difference of 1 oxygen is coming. If you look at the formula of chlorophyll A and chlorophyll B, because in chlorophyll A we have CH3 group, while in the case of chlorophyll B we have CHO group. So this 1 oxygen of the CHO group is causing the difference in the formula. Okay, so this is about the chlorophylls. Now the second type of a pigment that we have to study is carotenoids. So in the carotenoids, we saw there are 2 types of carotenoids. The first one is xanthophytes. The first type of carotenoid is xanthophylls. And what are xanthophylls? So the chemical formula of xanthophylls is C40H56O2. Okay. And these xanthophylls, they give yellow color. So these are yellow pigments. Okay. These gives yellow color. Now the second type of carotenoid is carotene. Carotene. And the chemical formula of the carotene is C40H56. Okay. So, and the color that it gives is yellowish orange. So it gives yellowish orange color. So this is all about the carotenoids. Now, how the scientists knew that the plants or the higher plants consist of only these pigments and not the others? Why does not, how did they know that it does not consist of chlorophyll, C, D, right? Phycobilin. So for this, so how does this authors know that what experiment was performed to identify the number of pigments that are present in plant leaves? So the experiment that was performed is known as paper chromatography. The experiment is known as paper chromatography. So what happens in the paper chromatography? So it is a chromatographic technique which what it does, it separates the various elements that are present in a compound, various components of a compound based on different characters. Okay. So in the paper chromatography, it is based on their mobility. Okay. So here what was done? Here there was the authors took a leaf structure. They took a leaf structure and then they crushed it to make a paste. Crushed it to make paste, and then they dip this in the paper chromatography. There comes a paper strip. Okay, so there comes a paper strip. So they make this paste. So they consist of this crushed leaf paste, and then they dip the paper strip in it. And what they observed 4 spots that was formed onto this paper strip, and these spots look something like this. Okay. So what happened here? What they are seeing there are various compounds that are present in this leaf. They move across this paper strip, and the compound that was lighter in weight that moved farther in that strip. So the compound that is present here at the higher, it is lighter one. It is lighter, while that was present at the bottom, it is heavier one. So in this, what they observed only 4 spots. Okay. And from this, they said that there are only 4 pigments that are present in the plant leaves. And these pigments in the order are, the first one is carotene. The second one is xanthophyll. The first one or the lighter one was the carotene. The second one was xanthophyll. Third one was chlorophyll A. And the fourth one was chlorophyll B. So the chlorophyll B is the heaviest one, while the carotene was the lightest one. So using this paper chromatography technique, the scientists determine that there are only 4 pigments that are present in the plant leaves, and that these pigments are involved in the photosynthesis. So we will stop our lecture today here. In the next class, we'll study about the light reaction and dark reaction. That's all for today. Thank you.